My name's Dr. James Volmer, and today with Dr. Pam Engelberts, we're going to give you a little bit of an insight into the research we're doing at the Center for Microbiome Research with Professor Jean Tyson in developing new approaches to understand the microbial world. So just a little bit of background, because Jean unfortunately couldn't be here today. The Center for Microbiome Research has several different groups and group leaders that have diverse expertise across microbiology. And this really gives us a unique opportunity for highly collaborative research. As I'm sure you're aware, Professor Jean Tyson has a wealth of multi-omic knowledge that he's used over the years, and he applies this across many projects within the center, with his specific projects focusing on the human microbiome. Associate Professor Simon McElroy focuses on wastewater and anaerobic methane oxidation, as well as the development of microbial visualization techniques. Associate Professor Ben Woodcroft is a computational biologist and focuses on the development of novel bioinformatic tools that he then applies to the novel datasets we have within the center. Professor Phil Pope focuses on microbiome animal-host interactions with a specific interest on ruminant and human microbiome. Associate Professor Luis Pedro Coelho focuses on the characterization of novel small microbial proteins and antimicrobial resistance. And Associate Professor Sonia Henriquez focuses on antimicrobial peptides and the characterization of novel cancer treatments. In addition to this, we also have close collaborations with Professor Greg Cook, Professor Yi-Chin To, Professor Tony Canna, and Dr. Mark Adams that provide us with additional expertise across infectious disease, bioengineering, immunology, and cancer microbiome interactions. So across the center, all of these groups work together on some key research themes that we have. The first is the human microbiome, where we're developing new strategies to treat diseases and improve human health. The second is the rumen microbiome, where we're developing new strategies to decrease methane emissions and increase animal productivity. Next is climate change, where we're investigating the impacts of climate change on climate-critical ecosystems, where we have multi-year projects looking at the effect of climate on the microbiome of specifically interesting sample types, such as permafrost and lake sediments. And as well as this, we're also looking at anaerobic methane oxidation, uncovering new lineages and mechanisms involved with methane consumption, and the wider role that these play in global methane cycling. For today's presentation, we're going to focus on three key aims that we have from the center. The first, I'll tell you a little bit of the bioinformatic tools that we're developing at the center and how they've been applied to the human gut microbiome. I'll show you some single cell approaches that we're developing specifically for our high throughput cultivation platform. And then lastly, we'll tell you a little bit about the novel visualizations approaches that we're developing and how they're being applied to decipher interactions between microbes of highly complex communities. So as I'm sure you're aware, over the years, Jean and his groups have developed a number of widely used bioinformatic tools. One of these most known groups are the M tools, which include CheckM1, which is used to give you a quality assessment and contamination assessment for metagenome assembled genomes or isolate genomes, as well as Single M, which gives you a comprehensive taxonomic profile of metagenomic data that Ben recently had published in Nature Biotechnology, which is a really good achievement. Dr. Alex Chislovsky, who is a postdoctoral bioinformatician at CMR, also recently published CheckM2, which improves on CheckM1 specifically by using machine learning for symbiotic lineages, which improves their predictions. As well as this, Ben and Dr. Sam Moroney, who's actually chairing this session, they've made a number of novel bioinformatic tools under the Aviary suite, including IBIS, also known as bin chicken. What's cool about this program is that it performs targeted co-assemblies across multiple different samples. This increases the number of representative genomes and quality of genomes that you recover. And Sam actually recently published this in Nature Methods, showing that he was able to recover the first representative genomes of thousands of new species, which is very cool. One novel bioinformatic tool that we're developing at the center that I want to highlight a little further today is Recurrem, being developed by Alex. So plasmids and other extrachromosomal elements are typically difficult to identify, and some of the existing methods used to identify them rely on databases of known plasmids, which ends up skewing your results towards those known plasmids. As well as this, assembly-based methods are often computationally intensive, and linear plasmids can be very elusive. So this is where Recurrem comes in. Recurrent works by identifying recurrently assembled contigs across multiple different sample types. Because you identify these recurrently assembled contigs across multiple samples, this gives you an additional level of high confidence that the things you are finding are in fact real and can be used to identify circular and linear elements. So using this tool that Alex developed, he then applied it to more than 10,000 human gut metagenomes, which resulted in over 9,000 circular elements and more than 34,000 linear elements. These elements were then all filtered through publicly available databases such as GDB to remove potentially chromosomal elements, and then mapped to publicly available plasmid and virus databases such as MVDB and IMGVR to annotate whether these elements were viruses or plasmids. From this, 5,500 were determined to be novel, with just over 500 being predicted as viruses, 1,800 as plasmids, and more than 3,000 as unknown mobile genetic elements. Collectively, these were then functionally annotated, and then co-abundance analysis was performed in an attempt to identify their host species as well. So this here shows just a little bit of information about the 1,800 novel plasmids that Alex recovered. So on the left-hand side here, this shows the size distribution for the plasmids. And as you can see, the majority of the plasmids fell within that 5 to 20 kb range, suggesting that they were quite small. In fact, most of these plasmids actually had only replication or toxin-antitoxin genes annotated, with the other majority of the genes being hypothetical or domains of unknown function. In saying this, you can also see towards the bottom of this graph that Alex was able to recover some quite large plasmids, even more than 200 kilobase pairs, which is significantly larger than average. On the right-hand side here, the main thing to see is that the vast majority of the plasmids Alex recovered actually only came from between 5 to 10 samples, suggesting that each one of us actually encodes for a unique plasmidome within our gut microbiome. So the next thing Alex wanted to do was to see if he could predict the host range of these plasmids that he recovered. He did this by using co-abundance analysis, but first wanted to validate that this was indeed a good approach. So he focused on known plasmids. What he did is he mapped the number of reads to either the plasmids or the host species, divided this by the total number of reads, and then input this result into FlashWeave, which is a program used for co-abundance analyses. This was then compared to publicly available isolate information in which the hosts and the plasmids were known. What Alex found was that the majority of predicted hosts that he was getting from his co-abundance analysis were confirmed by public host information, suggesting that this method was in fact a good way of identifying hosts for the novel plasmids he recovered as well. An example of this on the right-hand side here, you can see he recovered a Bacteroides fragilis plasmid that from his co-abundance analysis was actually associated with multiple different species of Bacteroides. Subsequently, he was also able to identify this plasmid in isolate genomes on RefSeq from other Bacteroides species, confirming that this was indeed a plasmid that was more broadly associated with Bacteroides. So using this method, he then applied this to identify the hosts of the novel plasmids that he recovered. In total, 210 novel plasmids were linked to more than 100 host species. This is for the 1,800 novel plasmids that he recovered, where more than 50% were found in greater than 5 samples. In addition to this, he also noticed that there were specific species that played a key hub role within certain families, like the UBA1417, you can see in the middle of the screen, within the Acutaliobacteriaceae. Within this family, 20 out of the 26 detected plasmids were associated with this species. And not only this, the plasmids were each unique and only associated with this species. So I hope today I've been able to show you what an interesting tool Recurrome is and how it can be applied to the human gut microbiome to recover many novel mobile genetic elements. In addition to the development of novel tools that we have at the center, we also focus on better characterizing the human microbiome through 4 main projects. First is our NHMRC Ideas Grant, which looks at characterizing the mucosal associated microbiome in inflammatory bowel disease. Second is our Defense Sciences and Technology Group grant, where we're looking at anxiety, depression, and cognitive performance and its association with the human gut microbiome. Third is Gene's ARC Laureate, which endeavors to produce a highly integrated multi-omic data set to provide a predictive understanding of the human microbiome. And then lastly, we're also characterizing novel drug delivery systems for targeting antimicrobial resistance and infection. One thing that's consistent across all of these different projects is the need for cultured isolates, not only to validate our bioinformatic findings, but also to better characterize these species in the lab. Unfortunately, at this stage, approximately 63% of the human gut microbiome remains uncultured. So there really is a critical need to be able to isolate these lineages of interest in a high throughput way. And that's where my work comes in. So at the Center for Microbiome Research, my main role was to develop a high throughput cultivation method that could be applied to the human microbiome to isolate not only these lineages of interest, but more broadly, all of these uncultured lineages within the human microbiome as well. And in order to do this, we started the Australian Human Microbiome Biobank, a platform and culture collection with 4 main aims. The first was to build and validate a novel high throughput cultivation platform that we could apply to the human microbiome. The second was to produce a collection of gut, skin, oral, and vaginal samples for microbial cultivation. The third was to produce genomic and predicted functional characterization for all of the isolates that we produce. And then lastly was to combine all of these isolates with their genomic and functional characterization into the Australian Human Microbiome Bank, which then we could apply to our various research projects, but then also provide to other researchers and industry. This was the basic workflow that we use. So we collect samples from volunteer donors and we process them fresh to maintain as much viability as possible. These samples are then homogenized and diluted and optionally stained with various DNA and metabolic activity stains, which allow us to target certain populations within the sample. These samples are then run on our fluorescence activated cell sorter that we have housed inside custom built anaerobic chambers. These anaerobic chambers allow us to change the gas composition inside. So if we're sorting a fecal sample, we'll obviously keep that largely anaerobic. But if we're sorting a skin sample, we'll introduce more oxygen to keep those aerobic organisms happy. Single cells are then sorted onto culture media, propagated out, and then all of the isolates we produce short-read sequencing whole genome data as well, really giving that kind of taxonomic and functional resolution to all of the isolates we produce. In addition to this, we also perform long-read sequencing on some specific isolates of interest to close out the genome. And then all of these isolates are then cryopreserved within our culture collection for future use in various research projects. So to date, AHMB has been highly successful in what it's sought out to do. We've recovered more than 25,000 isolates across fecal, vaginal, oral, and skin. As you can see by the pie chart in the bottom right, the vast majority of our culture collection is currently fecal isolates because that's the sample that we started with and we've spent the most time and optimized our workflows on. But since then, we've also expanded to our other body sites and we're seeing a lot of success in the number of isolates and the diversity we're recovering there as well. As you can see by the table on the left, we've been very successful in the diversity of isolates we've recovered from fecal samples. We've recovered both archaea and bacteria through our platform from 12 phyla, 14 classes, 30 orders, 60 families, 234 genera, and more than 650 unique species. The other thing you can also see here is that not only in terms of the number of species, but also we've been very successful in the previously uncultured lineages we're able to recover, with almost 40% of our species being brought into culture for the very first time, as well as several higher order taxa as well. When we compare AHMB to other recent culture collection efforts, you can also see that AHMB also stands out. So from the bar chart that's horizontal in red, this shows the overall size in terms of the number of species that each culture collection has, and AHMB has almost double the closest other culture collection. From the vertical bar charts here, this shows the number of unique species for each of the culture collections. And you can see that AHMB here almost has 300 unique species that are not found in any of these other recent culture collections. And I should preface here that all of these culture collections are from the human gut microbiome, not including the other species we've got from the other body sites as well. We've recovered the first isolates of many wanted lineages, such as the families CAG 272 and 274, as well as the Aristolaceae, which are getting more and more literature suggesting that they're associated with good health. In addition, we've also got many newly cultured species of known lineages of interest. I'm sure you guys have all heard before about Faecalibacterium or Faecalibacterium presnitzii specifically, and how it's a good gut bug and has anti-inflammatory properties. but we've got many lineages of this genus that have been cultured for the first time. So we're able to characterize those in the lab and see if they are similarly beneficial for human health. Not only this, but because we're undertaking such a large cultivation effort, we can also start to look at the strain diversity of certain species that we're recovering. The graph here on the left shows the proportion of the pangenome of these isolates, which is the core genome. And then on the y-axis, this is the number of representative genomes for each of the respective species. What you can see here is that for some specific species, such as the P. dorii and B. uniformis that you can see on the graph, as we're recovering more and more isolates, we're also increasing the genetic diversity of the isolates we have, to the point where some of these species, such as P. dorii, the proportion of their pangenome that makes up the core genome is actually only 10%, suggesting that they are extremely genetically diverse. And you can see on the right-hand side here that for P. dorii, only in the cultured isolates we have more than almost 25,000 unique genes, again showing that the isolates that we have are extremely metabolically diverse. If we look into this a little bit further, just as an example using P. dorii, I've included this graph on the left-hand side here. This graph shows each individual point as being a gene within the pangenome of P. dorii, where red are core genes, green are moderately prevalent genes, and blue are rare genes. As you move towards the right on the x-axis, these genes are more prevalent in our cultured isolates versus towards the left, which are more prevalent in the metagenomes. And then as you go up the y-axis, this is the overall abundance of the gene. What you can see from the P. dorii pangenome here, the vast majority of these genes within the pangenome are rare and more prevalently detected in our cultured isolates, suggesting that the cultured isolates we have provide essential genetic information that could be missed if you're just using metagenomics alone. In addition to this, we can also look at other things that these isolates encode, such as mobile genetic elements. So using Recur-M, which I mentioned earlier in the presentation, our culture collection, we've identified more than 170 different plasmids across a third of the species. The phylogenetic tree on the right-hand side here, this shows the diversity of isolates which have plasmids, with the bar charts on top in green representing the number of known plasmids for that given species, and the bar chart in red representing the novel plasmids for that species. Here, approximately 60% of the plasmids were determined to be novel according to Recur-M. Not only this, but because we've actually cultured these isolates, we know what the host range is of these plasmids. Using this information, we've identified multiple broad range plasmids, even across different phyla. And we further use this information to validate the co-abundance analysis that Alex performed and I mentioned earlier in the presentation. So I hope today, for my part of the presentation, I've been able to show you what an important resource AHMB is. We started this culture collection not only for our internal projects, but also to be a resource for industry and academic researchers all around the world. So if you have any questions, please reach out or scan the QR code on screen for more information. Charlotte is our AHMB coordinator and I'm on the research side of things, and we're always more than happy to talk about AHMB and any collaborative opportunities we have. From this point, I'm now going to hand over to Pam.
Thanks, James. So for the last part of this talk, I would like to highlight some of the work that we've been doing on developing novel methods to visualize microorganisms in complex communities, and in particular, GenomeFISH, which is genome-based fluorescence in situ hybridization for strain-level visualization of microbial communities. Now, for the last 3 decades, fluorescence in situ hybridization of fish has been an essential tool to visualize the spatial organizations of microorganisms. And fish detects microbes by the labeling of the 16S or 23S subunit of the ribosomes with fluorescently labeled probes. Now, to do this, you start off with any given sample that you fix the microbial cells and you permeabilize them to allow your fluorescent probes to enter. The next step of fish is hybridization, where you add your fish probes, and these are oligonucleotides of about 15 to 20 base pairs with a fluorophore attached to it that are reverse complementary to your target, which is the ribosomal RNA. Now, if you then design fish probes that target one microorganism, label them with one fluorophore, design fish probe for a different organism with a different fluorophore, you can start visualize your microbial community after you wash away any probes that don't bind. Now, fish has provided key insights into microbial morphology, relative abundance, as well as their co-localization, allowing you to start look into syntrophic interactions. And moreover, when you combine fish with stable isotope approaches, you can directly link the identity of a microorganism with its function in situ. And thus, fish can actually validate all the functional predictions that we make based on metagenomic approaches. But fish has a number of limitations. So first of all, its phylogenetic resolution. So due to the conserved nature of the ribosomes, which is your target, you can only reliably visualize microorganisms down to genus level, missing important species and strain level information. Secondly, your signal strength can be low. If you have a microbe that's not very active and it has very few ribosomes, then it has very few targets, so your signal can be hard to see. And then if your sample is also very autofluorescent, it could be human tissue, any given host tissue, soil, sediments, it's very challenging to visualize your microbe of interest. And lastly, fish is low throughput, and then it takes quite some time to design, synthesize, and optimize fish probes. So making it impractical to scale this method to the diversity of microbial life that's currently being discovered through metagenomic approaches. So there's a need for high throughput scalable approaches that can visualize microbial communities at strain level. And that's why we developed GenomeFish. And GenomeFish is a novel genome-based fish approach that couples high throughput single cell genomics with whole genome hybridization for strain level visualization of microbial communities. Now the workflow, you can start off with any given sample, environmental, clinical, whichever you're interested in. And then we use general DNA stains to stain the bacterial DNA, and then we use fluorescent activated cell sorting to individually sort single microbial cells into 96-well plate. We then lyse these cells, we amplify the DNA, and we send these DNA out for sequencing. We assemble the reads into single amplified genomes and identify the microbes. And then for microorganisms of interest, we take the amplified DNA and we fragment the entire genome to about 130 base pair fragments. And then we fluorescently label these fragments with chemically labeling kits. And these are GenomeFish polynucleotide probes. Now, they're very similar to fish probes, but in contrast to fish probes, you don't have to design them, and they target the entire genome instead of the ribosomes. So we can then hybridize these GenomeFish probes back to your original sample in a very similar way as standard fish. So we first optimize GenomeFish on a range of in-house cultured isolates. I'll show those results first. So here we see proof of principle of GenomeFish on an E. coli. So in the left, we have our GenomeFish signal. In the middle, we have traditional fish, because we make sure that GenomeFish is compatible with traditional fish. So we can visualize both strains, but also higher taxonomic levels. And then on the right, we have the overlay. So you can see that unlike traditional fish, where the signal is more distributed throughout the cell, targeting the ribosomes, the GenomeFish signal is more concentrated, targeting the genome. And this also allows you to visualize genome duplication prior to cell division, as indicated by the white arrows. Now we then apply GenomeFish to a whole range of microbes to show it doesn't just work on E. coli, but gram positives, gram negatives. And then we started mixing microorganisms to show the specificity of GenomeFish. So here we have a mock community of Pseudomonas aeruginosa in red, and then E. coli strain in green. So after showing proof of principle, our next question was, how far can we push the specificity of GenomeFish? Can we actually visualize strains? So for this, we ran an experiment where we collected the E. coli strain CFT073, which you can see on the left, and we generated GenomeFish probes for this strain and then applied it to a range of microbes with decreasing ANI values. So you can see we have a closely related E. coli strain, EDL, that has 97% ANI, a different Escherichia species, down to Pseudomonas aeruginosa on the right, which has 75% ANI. Now, the GenomeFish signal drops the more distantly related the organism, which is as expected, but it also binds to your closely related strain because the genome sequence is similar. Now, you can then imagine if you have a mixed community and you have a dimmer signal, how do you know if that's your off-target signal or maybe your target that is not fully in focal plane, or maybe it has lower genome copy number? Now, to account for this problem, what we do is we normalize to the DAPI signal. So for our target population on the left, our GenomeFish signal and DAPI signal will have a one-on-one correlation. While for a closely related strain, the GenomeFish signal will drop but the DAPI signal will stay strong. So if you normalize to DAPI, the value will drop below 1. And that is what you see here. So here we plotted the normalized signal intensity on the y-axis over average nucleotide identity on the x-axis. And in dark blue, you can see E. coli strain CFT with its own genofish probe, which has an average normalized signal intensity of 1. And this drops dramatically at the species level, but you can see that even for closely related strain, the signal intensity already drops. So it shows that genofish can be used to visualize strains up to 97% A9. And what does this look like in a more complex community? So here we have 2 E. coli strains, the CFT and EDL, an Escherichia fergusoni and a Pseudomonas aeruginosa. We applied 2 genofish probes, one targeting CFT in green and one targeting the E. coli EDL in red. And you can see they bind strongest to target, they bind less strong to off target, and not to Pseudomonas because it's too distantly related. And then when you overlay the signals, you can see that each of these microbes become visually distinguishable. And these 2 strains have 97% A9, but we push this up to strains that have 99% A9, so we show that we can use genofish to still distinguish strains with 99% A9. And linking back to the biobank that James mentioned earlier in the talk, it's super valuable to validate and run all these control experiments, but also he has hundreds of strains from the human gut that we can rapidly generate dozens of genofish probes for in days, where we can just extract the DNA of these strains and then fragment it, label it, and apply it back to any given sample of interest. So this could be your fecal sample or biopsy samples. But we want to make sure that genofish is also applicable to the uncultivated majority of microbes. So we applied it to 2 more complex environmental systems. Actually, the first one is an engineered system. So we applied it to a bioreactor community. And what we did is we ran the full cycle approach, where we stained the microbial cells, and then we sorted the single cells, and we recovered single amplified genomes, and then made genofish probes of single amplified genomes. We applied these genofish probes back to show morphologically distinct populations. So on the top left, we have an archaea, which does anaerobic oxidation of methane in this system and is known to form these clusters. Top right, we have a planktomycete. On the bottom left, we have a member of the IGNAV bacteria, and the bottom right, a Peteschi bacteria. Now, what we show in this system is that genofish can reach up to 27 times higher signal intensities than fish for tested species. As you target the entire genome with fluorescent probes, you get superior signal strength. And we can push this to visualizing species that have not been visualized before, where I mentioned the bottom right is a Peteschi bacteria, which is very challenging to visualize with standard fish, presumably due to its low number of ribosomes, but we were able to capture it with genofish. Now, the second system we applied it to is a human fecal sample, and this is to show the high throughput of genofish, where we rapidly isolated dozens of sacs and made genofish probes for. And here I'm just showing 6 of them that we applied back to the fecal sample to look at the sample at species level. So top left is Prevotella coprii, which was the most abundant. Bottom right, Phocaea colofulgatus, which was about 0.5% abundant. And then we can start combining species level probes that we label with different fluorophore to look at co-localization. And we've also pushed this to strain level, where we collected 2 different single amplified genomes of 2 strains of Agadobacter rectalis, and then applied the genofish probe to the fecal sample. So we have strain 1 in yellow and strain 2 in red. And these strains have up to 98% ANI. So this shows that even in more complex communities, genofish can reach strain level specificity. So to conclude, for genofish, compared to existing fish methods, genofish is high throughput, can reach superior signal strength and strain level resolution. And his work was published in the ISME journal last year, where it received the editor's choice, so feel free to have a look if you want to know more about it. Now, before I close, I'd like to highlight 2 more methods that we're developing at the center, and this work is done by 2 very talented PhD students. So we have Lara Carrington on the left, who's working on plasmid fish, and Harry Ling, who's working on virus fish. And they're very similar to genofish, but instead of targeting the host genome, they target either the plasmid or the virus. And they've both shown proof of principle. So on the left, we see an E. coli in blue with its plasmid in red, a 90 kb plasmid. And on the right, Ferry showed a viral infection of a single-stranded DNA phage that's infecting a Pseudomonas. And the Pseudomonas is in red, and the DNA phage is in green. So I'm sure you'll hear more about these methods in the future. But for now, I would like to thank you very much for listening. And I would like to thank the team, our colleagues. Without them, these projects would not be possible. Of course, the funding for the center and our supervisor, Gene Tyson. Without him, these projects would also not be possible. So thank him for our mentorship throughout.